All right, notebooks out, please. Our textbooks open. I'm gonna go through 1.6, so we will have the last chapter before our unit test. Today is Wednesday. On Friday, we will finish the lab, and next Wednesday will be the test. Maybe not. I'll give you some practice test day first, and then maybe Friday. But we're finishing today, okay? I I urge you not to ask me irrelevant questions, only powerful questions. And please remind me if I like tell you too many stories. Just tell me miss time, because I intend to finish quick because we're gonna start the lab. And mitosis is not that it's not that hard. Uh, the only two big ideas for you to know here is know the key events in the different stages of mitosis and know the concept of cancer. How does it happen? How is it not controlled? This is our big question. Why do cells need to divide? And as they divide, why does it need to be controlled? So let's learn cell division, mitosis. Are we in our listening mode now? Yes? All right. So the role of mitosis, as you can see here in the diagram, these are the different stages of mitosis. As else you have it in your notebook. The cell cycle is a cycle, so there's no start, there's no beginning. We don't really know where the cell is at one point, but we know the order. Interphase, and then you go mitosis, which is made up of prophase, metaphase, anaphase, telophase. And then you have cytokinesis, so it's division of the cytoplasm. So why do you think, I'm not gonna, maybe you know, what are the three reasons why cells need to divide? Yes. I see the answer in your eye. To?
Like, to grow and stuff.
For growth. Remember when I told you? Union of sperm and egg, that's one cell. What happens if you don't undergo mitosis? I'm going to be teaching one cell. I'm also one cell, teaching one cell. So for growth, the first one is embryonic development. So this is you. Look at you, so cute. You're so cute. That's us in the beginning of embryogenesis. Thank God for chemicals that signal the genes to be expressed, and you have different organs, different cells in the body. What else? Why do cells need to divide? We have growth. What else? Repair. So, for tissue repair, what do you mean repair? It's broken, right? How is it broken? Sorry, what? Oh, if you get a wound. Okay, if you get a wound, you have to repair those walls of your blood vessels. The walls are made up of cells. If you're exposed to the sun, your skins are damaged. If you ingest like toxic chemicals, parts of your organs can be damaged. So it's okay because the tissue can repair itself. The cells in the tissue can repair itself. Here's the problem. Not all cells of the body has the ability to undergo mitosis. So for example, skin. Skin cells has faster rate of mitosis compared to, let's say, liver cell. Once your liver cell is damaged, it's damaged forever. Once your brain cells are damaged forever. It's not like when I get Alzheimer's. Like, you know, Alzheimer's, that's hardening of the cells. I can't get new cells. I can't tell myself, go, mitosis, just give me more brain cells. So not all tissues can repair themselves. Is that clear? Only certain cells can undergo fast rate of cell division. Last, so growth, tissue repair. So out of my 22 students, nobody remembers from their reading. It's in your reading notes? I hope. For reproduction. For reproduction. So let's not think about us. Let's think about other organisms. Some lower organisms, they don't produce gametes, no sperms, no eggs, but they can still reproduce. So I have a picture here. Let's say this is a microscopic amoeba. So how does amoeba reproduce? To have more offsprings. So like just there's two amoeba. Isn't that wonderful? Like you want to have another of yourself. 2, 4. If you watch, have you watched The Monkey King? Ah, see, that stories again.
Makes me want to hear it.
No. So, some organisms use asexual reproduction to produce more cells, and they undergo mitosis. Okay. How many reasons do you need to know? 3 reasons. All right, let's look at the different stages of the cell cycle. This might be a review for many of you. I will emphasize key ideas per stage of the cell cycle. So, let's start with the interphase. So, in interphase, it's divided into 3 subparts, G1, S, G2. What do you think G stands for from reading? Growth or gap, right? Gap or growth. So at this gap stage, sit where you're supposed to sit later for the conduct card because I'm already lecturing. Open your notebook so you stay with me. You don't daydream. You doodle in your reading notes. Where's your notebook? William, see what I mean? See what I mean? Okay. All right. Gap or growth. So at this stage, the cell is actually very active. Why? It needs to grow. It needs to form. Remember last time you told me it needs to form more organelles. It needs to duplicate the DNA, and it needs to make more proteins. So specifically, let's look at each stage. G1, you make organelles, you make proteins, and you increase the size. I remember you answered that last time already. Why does size need to be increased? Because you're going to divide into 2. So the new cell should be about the same size as the original. Why do you need to make proteins? Are proteins important? The proteins have so many functions, right? One important function of proteins inside the cell is it acts as, I heard it, enzymes. So there's so many chemical reactions inside the cell. Chemical reactions need specific enzymes. So the cell needs to make lots of proteins. All right, S. S phase stands for senina. This is a very important stage of the cell cycle. Without the S phase, you cannot duplicate your DNA. And we just learned this, right? In 2.6, DNA replication, helicase, polymerase, please remember that. HLs, this should look very familiar. This involves all the enzymes you need to know, the topoisomerase, polymerase 3, polymerase 1. HLs are like, am I supposed to though? Okay, so the cell duplicates its DNA, again, to make sure that when it divides into 2, each cell will have the same, okay, nobody's listening to me, same DNA. G2, growth 2 or gap 2, the cell continues to grow, the cell continues to produce proteins, it continues to increase its cytoplasm. In other words, in interphase, please remember the most important part of interphase is S phase. Now, the cell is big, the cell has duplicated its DNA, the cell has more organelles that it needs. It will go into the next stage of the cell cycle, mitosis, PMAT. Am I safe to assume that you've memorized this order? PMAT? PMAT? Yes? Okay. All right. Before that, though, let's talk about supercoiling of your chromosome. Your DNA molecule is very, very long. If you look at this diagram, what's happening here is wrapping around histones. HLs, you should know this by now. Histones are 8 protein subunits, right? And then your DNA molecule can wrap around those histones. What does this lead into? If it keeps wrapping around histones, and then your histones coil. Supercoiling. So coil around histones, and then the histone molecules itself coils. So coil, supercoiling. Why do you want it to supercoil? Why do you want it compact? easier to
Separate.
separate. Okay. If it's too long, inside your nucleus you have 46 DNA molecules. Each molecule is representative of each chromosome. If it's too long and in anaphase they separate, they have to separate like from the Earth to the Moon because that's how long it is, right? You don't have space inside the cell, so it needs to be compact. So when you separate, it's shorter, and yeah, you get it, right? I should stop talking. Yes, I should stop talking. So supercoil to make sure that they're condensed, they're compact, and it's easier to split them in what stage? Anaphase. Okay. Now, parts of the chromosome you need to know. We will learn more about chromosomes and genes in our next unit. For now, please look at this parts of the chromosome. Right there in the middle, we call it the centromere. Central meaning central. And the centromere is made up of sister chromatids. What are these sister chromatids? This is made up of your duplicated chromosome. So imagine that's your DNA, and then you replicated it, semi-conservative. So you have two products, two strands, right? So that's the two strands. They're held together in the center called the centromere. Okay? I'll mention the relevance of the centromere in anaphase later on. All right, so prophase. Formation of your paired chromosome, so that's your duplicated DNA. What disappears? Nuclear membrane disappears. The spindle appears. So right there, spindle apparatus appears. You have polar bodies, it also appears. The nucleolus breaks down. Microtubules form. Don't, when you're studying this part of the stages of mitosis, please refer to the purple box in your book, like the key events. Okay, because I'm going to go fast through the stages right now. So nuclear membrane disappears, nucleolus disappear. So you have appearance of these centrioles and microtubules. These are proteins. Are you looking at the screen? These are proteins that are in charge of separating. So you need these polar bodies, you need the centrioles, you need the microtubules because it will pull your cell in opposite poles. All right, metaphase. Only one to remember. They line up at the equator. Actually, only two to remember. The other one is your spindle attaches to the centromere. So this protein right here, those strands of proteins need to attach to the centromere because that's the center. So when it pulls it apart, you know that you separated it evenly. Powerful question or no? Clarification, yes.
Our spindle fibers and microtubular fibers.
Yes, yes, yes. You can also, some textbook you will read microfilaments, microtubules, centrioles. They're all the same. They're all the proteins that aggregate to pull your cells in opposite poles. Good question. Anaphase. So we are separating the chromosomes. Once the chromosomes are separated, okay, hold on. How many chromosomes do you see here? 4. How many chromatids do you see here? 8, right? Because a chromosome is made up of a chromatid. To clarify, that this batch can understand easily, to clarify, once the chromosome separates, we don't call it a chromatid anymore. We call it, again, a chromosome. So you don't say 4 chromatids moves to the pole. Once separated, we don't call it chromatid, we call it chromosome. Okay. And then in telophase, your chromosomes have moved to the opposite poles. And then the nuclear membrane starts to appear. Remember that this appeared in what stage? Prophase. And then the chromosomes start to uncoil again. They start to relax. They're like, okay, relax. We've separated already. Unless it needs to separate again, it will supercoil. So it will uncoil, nuclear membrane will reappear, and then these spindle apparatus, microfilaments, microtubules will start to disappear. In other words, What disappeared in prophase appears in telophase. What appeared in prophase will disappear in telophase. Kind of like the opposite. All right, cytokinesis. We're discussing this as a separate stage. Please remember, cell cycle has three parts: interphase, mitosis, and cytokinesis. You might ask me, so why is cytokinesis not part of mitosis? Because there's different ways of cytokinesis depending on the type of cell. So we separate it as its own stage. So this is the final stage of your cell division, and you have to learn how is it different between animal cell and plant cell. So if you look at the animal cell, there's formation of a cleavage furrow. So you might smile right now because you think cleavage, yeah, whatever. That's how it looks like. There's a cleavage. There's a cleaving in the middle of your cell. And right here, proteins will be deposited. We call them, remember actin and myosin? Do you know that those are the proteins needed for muscle contraction? So the same proteins are deposited here. And then they can shorten. So if they shorten, it cleaves the cell into two. So I have contractile proteins here, actin and myosin, and it cleaves the cells into two. Very simple. Cleavage furrow brought to you by actin and myosin that shortens. It cleaves the cell. Now for plant cell, a little bit different. So for plant cells, step one, you have vesicles moving here at the equator. Okay? After they move here in the equator, they will fuse. This I need to draw. So they move here. Remember, the vesicle is made up of a plasma membrane too. Okay. So when this fuses, what's happening now? Eventually, I'll have a membrane here and a membrane on the other side. You get it? So vesicles will move at the equator. They will fuse. So these two cells, so one cell here and then another cell here, will have their own separate plasma membrane. Yes, you understand, but you didn't draw, so you will forget. Second, please draw like this so you will remember. Second, another group of vesicles will, I need a book. Another group of vesicles will deposit two proteins. What are the two proteins? First one to see it, please. Pectin and cellulose. Okay? So we have another group of cells, group of vesicles that will release pectin. And it will also release, what's the other one? Cellulose. So pectin and cellulose will be deposited here in the equator. Pectin and cellulose are long strands, and they are, where did we learn cellulose? Alpha 1,4 or beta 1,4? I actually don't remember, please remind me. Cellulose. Cellulose is either amylose or amylopectin. Alpha 1,4 and alpha 1,6. HLs, please be sad if you don't remember. Because by now. Cellulose is beta. Okay. HLs, cellulose is beta 1 4. Starch, which can either be amylose or amylopectin, is alpha 1 4 alpha 1 6. I don't remember too. I don't remember. It's okay. I'm not taking the IB exam. Yes. I don't know. Okay. Where was it? You should know by now. You know that song? You ought to know by now. You have to know by now. I don't know. You should know. I'm not taking that. Beta up, alpha down. Okay. Beta up, alpha down. That was your report, Daniela, right? All right. So these two deposits of pectin and cellulose will become, can you guess? That's such an easy question. The cell wall. So in between your cells, you have one cell wall here, another cell wall here. All right. For the test, please don't draw for me. I need explanation. The question will ask you differentiate between cytokinesis of your animal cell and plant cell. Okay? Okay. We're almost done. Two more ideas. Cyclins and cancer. Let's talk about cyclins. So cyclins excuse me. Let me start with that one picture there. Discovery of cyclins. Who discovered cyclins?
Some guy whose name is
Some guy who's in your book, but let me know if you're reading your book. Who's this guy? Huh? Tim Hunt. He discovered this by serendipity, like not really planning on discovering this. So this, what is this? Sea urchin. So the sea urchin, as it reproduces, he kept getting water samples, and what he saw was there's kind of a trend of increase in cyclin, then decrease in cyclin, and it coincided with the stages of division of your sea urchin. So what can be the only conclusion? Huh? It cell division uses signals from the cyclin to proceed from one stage to another. This is new in your batch, your curriculum. So this is the same graph from your book. Can you please open to that? And page 56, okay. What you need to know for the test is which cyclin affects which progress in the cell cycle. So you have cyclin D, E, A, and B. Out of the 22, I have 6 looking at the textbook. So cyclin D, can you please tell yourself, I need to memorize this. Cyclin D triggers movement from GO to G1, GO to S. In other words, the whole interface. Cyclin E seems to trigger DNA replication in S phase. Cyclin A, some of you are writing in your notebook, very good. Cyclin A activates DNA in S phase. Cyclin B, where's my cyclin B? Red. Triggers the movement into mitosis. Okay. Now, how does cyclins work? This is just a hypothesis. So what they say is in the presence of cyclin, enzymes called cyclin-dependent kinases. We've mentioned kinase before. Remember? And then I said, remind me if I say kinase. You remember? I mentioned kinase somewhere. No? Oh. Okay. So cyclin right here. In the presence of cyclin, this cyclin-dependent kinase is activated. It's activated to affect assembly of other proteins needed by the cell. Why do we need proteins again? Proteins are your end enzymes to take charge of different chemical reactions. Now, these enzymes can only be activated if it's triggered by your CDK. And your CDK is only activated upon binding with your cyclin. So 3 steps. Step 1, cyclin binds to CDK. Step 2, CDK, what did I say? Oh, the word that I used. CDK, the assembly of proteins. Not catalysis. Triggers the assembly of proteins that activates different kinds of enzymes. Then step 3, these enzymes are now used for the different stages of mitosis. Clear? You're like, yeah, clear, Miss, but I don't want to remember them because it's not interesting.
But can you repeat the last one?
The enzymes, from the beginning. Cyclin binds to CDK. CDK triggers the assembly of proteins and activates specific enzymes. Step 3, specific enzymes are used in specific stages of mitosis or specific stages of the cell cycle. Yes?
For instance, it's the phosphate that triggers it,
So what's a chromosome?
or it's the
Just think activated enzyme. You don't need to know details. This is just a picture. It might look different, it's not really the phosphate. Just think this is activated enzyme because of these proteins, which is stimulated by this CDK, which is stimulated by cyclin. Just a model. But we really don't know if it happens this way. Although we really know that specific cyclins seem to increase in their concentration in different stages of mitosis. Yes.
So kinase is
It's an enzyme. It's an enzyme that activates proteins that activates specific enzymes. In HLs, remember our topic on metabolism? When you talk about chemical reaction, it's not from A to B. Remember the diagram that I showed? There's so many chemical reactions to get into your final product. All right, let's talk about cancer. Tumor information and cancer. Woo. Some vocabs, this is in your vocabulary sheet, right? Benign and malignant. So benign, there's an abnormal growth of your cell. It can be in any place of your body. But this is not yet cancerous. We call it a tumor. If it starts to keep increasing in size, because sometimes your tumor, it grows big and then that's it. It doesn't proceed on increasing in the size. Then that's just like a tumor, a benign tumor. But then the doctor will still need to keep checking that at least 4 times a year to see whether it's increasing in size. Like my sister has a lump in her breast. So the doctor said, we'll just have to keep monitoring. We have annual physical exam to check if the size is getting bigger. If not, then it's a tumor, but it's benign, meaning it's not going to ever become a cancerous tumor. It's just, in Tagalog, it's just a bukol, lump in the body. But, just random lumps. Stories again, no. If it keeps increasing in size, so you get checked up every year and then it seems to be increasing in size, then probably it's not benign, it's malignant. It means it keeps growing. And that is when we call it cancer. A malignant tumor is when you have cancer. Okay, just one quick story. So I had a lump in my body. I'm not going to tell you where. So there's a lump. And I'm so paranoid. I'm like, oh, is this cancer? Because it feels hard, whatever. And it's painful. So I got checked in St. Luke's and the doctor checked it. And then I'm like, doctor, is it just lymph nodes? I want to confirm what I think what it is. Is it just lymph nodes? And then the doctor said, without hesitation, the doctor said, it can be a lymph node or a tumor or an infection. And I'm like, lymph node, tumor. So far, doc. You could have said lymph node, infection, just maybe tumor. When I heard the word tumor, I'm like, mommy, lymph node tumor. I mean, my choices are it's a lymph node infection or it's tumor. Like, don't I have any choice in the middle? So the whole weekend, I was just staring blankly and then thinking about my life. And I'm like, oh, I'm not going to live anymore. He gave me antibiotic. He said, come back after 1 week. After 1 week, no more lump. So I'm like, oh, a tumor. It's not a tumor, doc. It's just a swollen lymph node. But anyway, so if you see lumps around your body, yes. Oh, lymph node. Let me. This is a system that is rarely looked at, but a lymphatic system is a series of vessels, not vessels because it's not blood. It's a series of tubes around the body, as you can see here, and it runs side by side your blood vessels. And in your lymph nodes, like this is your lymphatic system, your lymph nodes, that's where you deposit cell debris, or if the white blood cells, if you have a lot of white blood cells and they die, that's where they go. If you have infections in this area, the lymph nodes here swells and it's painful. If you have an injury here, the lymph node here swells and it's also painful. Have you ever had those? Like when your lymph nodes are swelling, the doctor goes like this, you palpate, and it's like a lump here. A lump here, it can be here in your inguinal area also, or here in the back of your feet. That's just lymph node. Actually, sometimes I get that if you have infection here, upper shoulder. But, yes, when it swells, like really big, yes. Mumps. But mumps are caused by viruses. And then it swells that lymph node. Okay, going back.
What if we don't cure what
For the lymph nodes, So I was thinking I had swollen lymph nodes, but then I said that's not good because if you have swollen lymph nodes, you can also get lymphoma, non-Hodgkin's lymphoma. That's when your lymph nodes are overacting and it's swollen. Like the cells of the lymph nodes themselves keep dividing. Lymphoma is cancer of the lymph nodes. So I'm like, I hate myself too much because I know so many things and it makes me so paranoid. Like, oh, I wish I didn't know anything, so I just know there's a lump there. Going back. Let's go back. Cancer. All right. 3 important vocabs you need to know. Mutagens. Mutagens are... You're not listening anymore. It makes me sad. Agents, it can be chemical, it can be environmental, that can change the sequence of your DNA. Carcinogen. This is not in order. Carcinogen are example mutagens that are specific on mutating your oncogenes. What are oncogenes? Those are specific portions of your DNA that are in charge of controlling cell division. Again, mutagens, any agent that can mutate your DNA. What do you call mutagens specific to oncogenes? Carcinogens. What are oncogenes? In other words, we all have genes to develop cancer. We all have oncogenes. But not all of us are exposed to carcinogens. Get it? And just this morning, story again, sorry. 10 minutes. Just this morning, I was reading the FDA announces that poultry or chicken has high levels of arsenic. Arsenic are mutagens, carcinogens. So there's a lot of comments, oh, I don't want to eat chicken anymore, blah, blah, blah. So I kept reading. They actually first investigated this in 2011. That's like 7 years ago, or 7 to 8 years ago already. Those people who are pro-chicken, pro-poultry, they kept saying, yes, it has arsenic, but it's in low levels. But then on the other side of the story, okay, even if it's in low levels, just the idea that there is arsenic in chicken, wouldn't that make you a little paranoid? Like you're consuming lots of these poultry, lots of these chicken. It made me think, should I buy in the grocery, should I buy, in Royal they have imported meat. I'm like, it's from the States. Oh, maybe not. I'm just gonna buy local poultry, chicken. Anyway, I was just think about all of the food that you're eating. You don't know how these foods are manufactured. That's why a good number of people are turning to organic food. They want their food fresh and organic. Because the way it was prepared, you don't know what chemicals they put. Also, your pork and your chicken, what are they fed? Are they fed healthy food? GMOs or feeds, chemicals. They're fed chemicals. Where does these chemicals, it's incorporated in their tissue. What do we eat? We eat their meat. So is it possible all of those chemicals will get into our body? Yes, that's why we have a lot of carcinogens. Because you don't know it, but you're exposed to carcinogens already. Yes.
If we cook it, does it disappear?
If you cook it, what disappears are bacteria. But then it's a chemical. So we don't really know. That's why there should be strict regulations. In the US, they have like the FDA, they try to screen all of their produce with all of the possible carcinogens. I don't know in Korea. Here in the Philippines, we have BFAD. But it's something we cannot control. I mean, it's so large scale that you can't really pinpoint the source or the cause of that cancer. But one thing for sure is that we are exposed to chemicals, for sure. Okay, enough. Continue. These are some example chemicals. Too much UV exposure. You've heard of skin cancer. X-rays, miss. Yeah, those are radiations. Those MRI, they sometimes if the radiation is too strong, it can change the sequence of your DNA. Then maybe it can lead to expression of your oncogenes. Carcinogens, smokers. I'm gonna talk more about smoking. I hope there's not a lot of smokers here. Processed food, again, because we don't know what chemicals are in your food. Cosmetic. I've read something, but there's no clear link. Like when you put makeup, sometimes you ingest the lipstick, right? You swallow the chemicals. Yeah, by accident.
You don't eat that.
Yeah, but chemicals are still getting inside your body. So there's like a specific type of lipstick that uses, what do you call that? What's the term? Like you can ingest kind of chemical. And you know how like makeup from, I'm not racist, I'm not stereotyping, but makeup from China, they put like whatever there in their chemicals. So you are exposed to these chemicals, even beauty products, the things that you put in your skin, it can get absorbed to your blood. So all the chemicals that you expose your body to. Okay, viruses and whatever. But we will focus right now on smoking. This is just a picture of your tumor forming in your tissues. All right, does smoking cause cancer? What's the answer?
Yes.
No,
No?
it doesn't cause cancer. It's correlated to developing cancer. If it's the cause, then if we all stop smoking, there's no more cancer in the world. It's just correlated. Look at the data. Just to convince you that they're correlated. Lung cancer, this is the mortality rate, number of deaths per year. 1768. That's about 30%. How many percent increase? 300.
400.
For about 400% increase. And then let's look at the other cancers. Cancer of the mouth, pharynx, look here. About 300. All other cancers. So incidence, just focus here. Incidence of mortality rate for cancer is higher among cigarette smokers. More epidemiological studies needs to be conducted. But so far, we have seen a link, a correlation between the two. Um, Although I'm not saying cost because I know of so many people that are heavy smokers, and they live until 80 or 90 years old. So what could it be if it's not smoking? If it's not the chemicals?
Maybe you keep ingesting while you
Chemicals or smoke. Well, what can be another factor if it's not all of these carcinogens? What's the next factor that you can think of? Genes, genetic. It may be genetic. I emphasized that before again. If you have it in your family, if you, I was talking to this friend who has a friend, instead of trying to look at the cost, this person mapped their family tree, then looked at how many people had this specific type of cancer. So this person said, okay, so many in my great whatever. So it means I may have a good chance of getting this specific type of cancer. So this person is trying to avoid, no, not avoid. This person is trying to do lifestyle changes because it's so obvious that if it's in your family, there's a good chance you can also get it. Yay, I'm done. Any questions with 1.6? We're going to do a lab now. When I say brain break, please be quiet. Just rest your mind. Rest your brain.
so